Hello and welcome to October 2023's Trache Talk. This is our monthly review of the literature that's caught my eye in the tracheostomy press. There's more resources on the website and companion documents to accompany this podcast if you want to take a look at tracheostomy.org.uk. But we're going to cover a few different papers this time that focus on everything from new surgical techniques, models for self-management at home, and improving the design of tracheostomy tubes. Hopefully essential listening for anyone involved in multidisciplinary tracheostomy care. Our first paper is from Bouchon in Korea. It's published in Clinical Experimental Otorhinolaryngology. It's a comparison between real-time ultrasound-guided PDT and surgical tracheostomy, all in critically ill patients. This is a randomised controlled trial, and the authors note this is the first time that this has been a randomised controlled trial to compare these two procedures. So the two main ways of getting a tracheostomy in are with an open surgical technique where you use a knife and a surgeon, and usually an operating theatre, to dissect the tissues of the neck and to get down to the windpipe, and you cut a hole in the windpipe and pop the tracheostomy tube in. Or a percutaneous technique, or a PDT, where you put a needle into the windpipe and then you use a succession of dilators to stretch everything up before you put the tracheostomy in. Now, ultrasound can be used to try and help place the needle in the right place. Ultrasound has many advantages. It can certainly help find the trachea in the neck. It can help to identify blood vessels that you don't want to hit with a needle. But what they're doing in this paper is using ultrasound in real time to guide the needle into the trachea. We've discussed this before on this podcast, that ultrasound has a few limitations, and I think if it was that good, we'd all be using it in intensive care practice. But trying to visualise on ultrasound the air-filled trachea is quite hard. The other major problem you've got is that you can often see the needle, but you don't know whether it's the tip of the needle you're looking at or some other part of the needle. And specialist needles are available, but typically not for PDT. But I think at the moment it's still a bit hard to use ultrasound for mainstream if you're not highly trained, highly specialist, and you've got really good kit, which certainly if you work in the public sector in the United Kingdom is not always the case. Anyway, back to the study. This is what the authors found. So the ultrasound PDT procedure time was significantly shorter than the open surgical theatre time for putting a tracheostomy in. So they had an average time of 4 minutes compared to 10 minutes for the surgical procedure. So even when you counted in the total duration of the whole procedure, which is positioning the patient, fiddling with the tube, and then adding the ultrasound in, it was significantly short of the ultrasound-guided PDT. Now that might just be that PDT is quicker than surgical tracheostomy, but certainly what they found was adding in this extra layer of complexity with the ultrasound, it didn't create any additional problems. They found similar complication rates. So the incidence of complications was almost identical between the two groups, and there was no major complications in either group. The PDT group had smaller incision, which is something I think is an advantage, particularly in the critically ill, which may lead to less problems afterwards, such as tube displacement and certainly wound infection. And the authors concluded quite reasonably that ultrasound-guided PDT is safe and it's effective potential alternative to surgical tracheostomy, with the advantages as they described, a shorter procedure time and a smaller incision. So I think this is another useful study which shows us that PDT is probably pretty good when compared to an open surgical approach for patients in the ITU. Ultrasound guidance may be the answer to help us place the needle very carefully, but there are other approaches, and if you have a look at the NTSP website, you'll see some work we're doing around trying to accurately insert needles with a really high degree of precision. There will always be patients who need surgical tracheostomy due to abnormal neck anatomy or to blood vessels in the way, and so that will always still be a place for surgical tracheostomy in our critically ill patients. But this is an interesting paper showing us that ultrasound-guided PDT is safe and effective. Our next paper is looking at biofilm in tracheostomy tubes. So most tracheostomy tubes have an inner cannula that you can remove, clean and put back in, which saves you having to remove the whole tracheostomy tube to clean it. And tracheostomy tubes get gunked up. They're in the airway, it's warm, it's wet, your airway and your lungs and your trachea are full of bugs and sputum and liquid and snot and all sorts of things that when they hit plastic will stick to it. And anyone who's looked after anyone with a tracheostomy will know that you need to keep that tube clear. The inner cannula are susceptible to something called a biofilm. And a biofilm is a complex, self-produced community of microorganisms, so things like bacteria, fungi or algae, and they stick to the surface. They encase themselves like a protective, slimy sort of matrix. And this sticky stuff is made up of things like polymers, polysaccharides, proteins and extracellular DNA. And so you get this sticky layer on the inside of the plastic inner tubes. There's some evidence that microorganisms in that biofilm communicate with each other. There's a process called quorum sensing, and that supposedly allows this community of bacteria living on the inside of these bits of plastic to coordinate its activities. They share nutrients, they shelter within that protective matrix. That means that these biofilms are often more tolerant to antibiotics, disinfectants, and the host's immune system than other microbes that might be floating around outside of that protective biofilm. So the authors feel that biofilms are a significant cause of morbidity in patients with indwelling medical devices, such as tracheostomies. And the particular use case here of reusable inner cannula potentially increases the risk of infection. The way we try and mitigate that is through decontamination, and that reduces what's called the bioburden, the amount of bugs living in this matrix. But the current guidelines for cleaning in a cannula are pretty varied. There's multiple techniques being recommended and they're not necessarily supported by strong evidence. And so this study asked a fairly simple question: what is the best cleaning agent for reusable tracheostomy in a cannula to reduce the bacterial load and therefore hopefully reduce the risk of infection? So this is a randomised control crossover study. That means that they randomised people to undergo two different cleaning regimes in different orders. So you had treatment A, then treatment B, or treatment B, then treatment A. And then what they wanted to do was work out what was the bacterial count on these inner linings. They took 60 patients and what they did was they used detergent or sterile water. Patients were randomly assigned to that sequence where they had water cleaning and then detergent cleaning as the regime, or vice versa. The inner cannula were flushed onto sort of agar plates, which are the standard things you'll see for growing bacteria in hospital labs, and they looked at what they grew. The key findings was that there was a high initial bacterial load. The pre-decontamination inner cannula showed very high counts, highlighting that yes, we do need to do effective cleaning. Water and detergents were pretty much the same at reducing the bacterial load for the safe reuse of inner cannula. They were doing something called a non-inferiority study where they're looking to not show that one was better than the other, but show that sterile water was no worse than detergents. So that meant that they were both microbiologically effective. Now the implications of this is cost savings, because sterile water is more expensive than non-sterile water, but it's still pretty cheap compared to detergents and specialist cleaning solution. So this has the potential to promote significant cost savings for tracheostomy patients and caregivers in both the hospital and community settings without compromising patient safety. So if you're involved in cleaning tracheostomy tubes and you're worried about infections, I think this is a neat little study that shows us that actually sterile water is not bad at keeping these inner cannula clean. The next study is another lab study. We're looking at residual above cuff volume when different tubes have been used with subglottic suction ports. This is from Katsuno and colleagues at the University of Tokyo in Japan. And what they did, they used cuffed tracheostomy tubes, and they had a variety of different brands of tube, a variety of different subglottic suction ports, and they basically worked out how effective they were at cleaning rubbish from above the subglottic suction port. Now, we use subglottic suction ports a lot in critical care, and the theory behind them is that if you have a cuffed tube in your trachea and your swallowing isn't that great, material that ends up aspirated through your vocal cords and goes into your windpipe gets caught above the cuff. If you leave the material there, some of it inevitably will end up in your lungs. And so the subglottic suction port is designed to remove that material that collects above the cuff. And the authors comment that removing that aspirated material is crucial to prevent complications such as aspiration pneumonia. And certainly it's a standard of care in the settings I work in, where we look at package of care to reduce pneumonias, and one of those elements of that package is subglottic suction tracheal tubes, which include tracheostomy tubes. So what they wanted to do was try in the lab different liquid viscosities, i.e. thick or thin stuff, different positioning of the ports, angles of the tilt on the test tube, and see how much volume was left above the cuff. So they used 5 different tracheostomy tubes. They were all about 9mm in outer diameter. They put them in a transparent cylinder which had an internal diameter of 18mm. So trying to replicate the human trachea, but clearly the human trachea is not a solid cylinder. It's not 18mm wide all the way down, and it's not made of see-through plastic. They inflated the cuff to completely seal the cylinder, and then they used 4 different liquids of different viscosities poured onto the cuff. They tried to then suction out the liquid above the cuff and see how much material was left and when they'd finished doing the subglottic suction. They then angled the cylinder at 90 degrees and 20 degrees to see if they could alter the amount of material that was aspirated. And again, if you think about that in real life, patients move around quite a bit, and so we don't stay completely vertical when we are trying to drain these subglottic suctions. So the key findings here was that residue was universal, and by that they meant that some liquid was observed on all the cuffs of all the tested tracheostomy tubes, even those with side ports suctioning low viscosity liquid. And this confirms the clinical reality that complete clearance of this material is really hard. Viscosity, though, and distance of the subglottic suction port from the cuff were the key factors. They found that the higher the viscosity of the liquid, so that the thicker it was, the harder it was to remove. And tubes that had a long distance from the subglottic suction port opening to the top of the cuff ended up with more residual fluid above the cuff. They felt that positioning of the subglottic suction port also was relevant. So if you put a subglottic port on the rear, on the posterior aspect of the tube, then when you tilted the tube, simulating a reclining position, then the amount of residual fluid there actually decreased. So putting the subglottic suction port on the back of the tube may have some advantage if the patient is in a reclining position. Tubes with a lateral port didn't show any difference in change of the angle. The authors have made some suggestions, and they suggest an optimal tube for drainage would have a posterior suction port and a short distance between the port opening and the cuff top. So if you imagine your tracheostomy tube, it's got a cuff, and immediately above the cuff at the back is the exit of the subglottic suction port, or possibly the entrance, depending on which way you are blowing or sucking material out. So, as I said before, these cuffs probably behave very differently in the warm, mobile human body. But it's a very interesting study nonetheless. It does raise some questions to me about the port positioning for therapies like above cuff vocalization, because we borrow the subglottic suction port and we use it to blow gas backwards. And I suspect that having a subglottic suction port higher up the tube, further away from the cuff, will be better for vocalization, but probably then worse for secretion aspiration. So there are also some systems that flush the subglottic space, which are available on the market, and they actually may be advantageous. There may be some related material out there that we'll cover on one of our other podcasts. But I think for now, this is an interesting study that again reminds us that all tubes aren't equal, and even though you've got a subglottic suction port in, it doesn't necessarily mean that you're going to keep all those secretions out of the way. Our last paper this time comes from Weidlich and colleagues from Freiburg in Germany. This is all about self-management in the home setting, and it's a scoping review, and they aim to systematically synthesize this model for complex self-management, and they've used this framework which is adapted from the comprehensive Lorig and Holman self-management model, and they've applied it to the specific population. So the methods behind this are certainly not my area of expertise. I would encourage you to go and have a look at the paper if this is the sort of thing that interests you. But I'll tell you what the authors found. They described 3 dimensions of self-management, and there were 3 overlapping dimensions. Management of a therapeutic regime, for example, tracheostomy care, dealing with an altered airway, dealing with altered speech, swallowing and breathing. Managing role and behaviour changes, which is altered self-identity, social participation and your professional life. And then managing emotions, so dealing with negative emotions like fear, depression and stigma, but also positive emotions like a life-saving feeling and then optimism for the future. What the authors describe is that participants and the research explain there's an emphasis on lifestyle over technical care. So while technical tracheostomy care is essential for safety, the majority of the literature that they looked at focused on the managing of the physical and functional changes and then the role and behaviour changes that you need to go through associated with living with a tracheostomy. So I thought that was quite an interesting thing that, often as healthcare providers we focus on the technical aspects, but actually there's a huge desire from our patients and families for this sort of lifestyle advice, lifestyle sort of focus, because that is arguably more important than some of the technical stuff. There were some day-to-day challenges highlighted, such as changes in social eating, communication, leisure activities, and they were cited as more important than the physical management after discharge. So the authors felt that this model serves as a foundation to guide multi-professional intervention studies to better support patients' complex needs when they're discharged from hospital into the home setting. So if you're involved in discharge of patients to home, I'd encourage you to have a look at this paper because it does highlight some really important themes that are very relevant to our papers. So these 4 papers have given us a pretty good snapshot of current efforts in tracheostomy safety and care. We've gone from optimising the procedure itself with ultrasound, thinking about inner cannula care, to a very detailed analysis of suction, and then we've finally ended up looking at a patient-centred model for preparing for discharge home. Hopefully this update will help inform your practice and your discussions. And that's it for this month. Thanks for listening to the end. We welcome any comments on our social media platforms, and as ever we'll try our best to respond to them. There's lots more information on the website, which is tracheostomy.org.uk. More podcasts, more videos, lots of information. Please note that the comments you've just heard are my opinions. They're not necessarily the opinions of any of the organisations for which I work. Thanks for making it to the end, and I look forward to seeing you next time. Bye for now.